Welcome to The Explainer. Today, we're basically going to be detectives. We're diving into a genetic mystery that starts in a quiet monastery garden and ends up unlocking the secrets hidden deep inside our own cells. We're going to follow the clues and see how a brilliant idea about heredity became a physical reality.
So where do we even start? Well, we have to go back to the guy who wrote the book on genetics, Gregor Mendel. He was the one who figured out the rules of the game, the patterns of how traits get passed down. But there was a huge question mark left hanging. Where in the cell was all this information actually written down?
Okay, so let's talk about these factors of his. Mendel was a genius. He saw the patterns, he understood the rules of inheritance, but he was missing a critical piece of evidence. He couldn't physically point to what these factors were.
Through his incredibly careful work with pea plants, he figured all this out. He knew there had to be these little discrete packets of information being passed from parent to child. Today, of course, we know them as genes, and their different versions as alleles.
And this is the real kicker. His ideas, as revolutionary as they were totally abstract. It was all on paper. He had the how, but he had no clue about the what or the where. He had the blueprint, but no idea where the building was. And so, just like in a classic detective novel, the case went cold for decades. But then, a new piece of technology comes along, better microscopes. And suddenly, we got our first big lead. Scientists could finally see inside the cell with incredible new detail.
Can you believe it? It took nearly 40 years for science to catch up. But right around 1900, Mendel's work is rediscovered, and it's happening at the exact same time that biologists are staring in awe at these mysterious thread-like things they called chromosomes, dancing around inside dividing cells.
So enter our lead detectives, Walter Sutton and Theodore Boveri. These guys looked at Mendel's abstract rules on one side, and then they looked at the physical behavior of chromosomes on the other, and they saw something that made the hair on their arms stand up.
And this is where it all clicks. The parallel is just perfect. Mendel said his factors come in pairs. Well, guess what? Chromosomes come in pairs. Mendel said factors separate when you make sperm and eggs. Chromosomes do the exact same thing during meiosis. The behavior was a mirror image. This couldn't be a coincidence. No way.
Right, so it wasn't a coincidence. This was the moment. This was the solution to that big mystery of where Mendel's factors were hiding all this time.
Sutton and Boveri put 2 and 2 together. They proposed that Mendel's factors, his genes, were physically located on these chromosomes. And just like that, we have the chromosomal theory of inheritance. It gave a physical home, a real address to Mendel's abstract rules.
So, case closed, right? We found the culprit, we know where the genes are. Perfect. But you know how these stories go. Just when you think it's all wrapped up with a neat little bow, a new twist comes along that threatens to unravel everything.
All right, let's bring in our next investigator, Thomas Hunt Morgan. And his lab wasn't filled with quiet pea plants. Nope, he was working with something a little more buzzy, fruit flies. And his experiments were about to throw a real wrench in the works.
So Morgan starts doing experiments similar to Mendel's, tracking 2 traits at a time in his fruit flies. And based on everything they knew, he fully expected to see that classic 9:3 to 3:1 ratio. That specific ratio is the mathematical signature that tells you 2 traits are being inherited totally independently of each other.
But that is not what he saw. Not even close. Instead, what he found was that the original combinations from the parents were inherited together almost all the time. The new mixed-up combinations were incredibly rare. It was as if certain genes were handcuffed together.
This was a huge problem. His results were a flagrant violation of Mendel's law of independent assortment. So what on earth was going on? If genes were on chromosomes and chromosomes sorted independently, why weren't these genes following the rules?
Now, you might think these confusing results would just tear the chromosomal theory to shreds. But what Morgan figured out next didn't just save the theory, it made it even more powerful. His first big idea was something he called linkage, and it's actually pretty intuitive. If 2 genes are physically located on the same chromosome, well, they're linked. Think of them as passengers on the same bus. They're probably going to get off at the same stop, right? They tend to be inherited as a single unit. Okay, so linkage explains why some genes stick together. But what about those few rare flies that had the new, unexpected combinations? For that, Morgan came up with his second brilliant idea, recombination. So even if genes are on the same bus, every once in a while, they can actually swap places with genes on another bus.
And this happens through a real physical process called crossing over. When the paired chromosomes line up during meiosis, they can literally get tangled up, break, and swap segments of DNA. It's the mechanism that shuffles the genetic deck, creating those new combinations that Morgan was seeing in that small fraction of his flies.
But then they discovered something truly incredible. The frequency of this crossing over, this recombination, depended on how far apart the genes were on the chromosome. Genes that were close neighbors almost never got separated. Genes at opposite ends of the chromosome got separated much more often. That recombination rate was like a ruler. They could use it to actually map the location of genes on a chromosome.
So, you see, linkage and recombination weren't holes in the chromosomal theory. They were the missing chapters. They explained the exceptions to the rule, and in doing so, they gave us a much deeper, more sophisticated picture of how inheritance really works. The case was finally, truly closed.
And just think about where this led. One of Morgan's students, Alfred Sturtevant, used this very idea to create the first ever map of a chromosome, all by just counting flies. That basic principle laid the foundation for everything that followed, including the monumental Human Genome Project. It all started with a few unexpected results in a bottle of fruit flies, forever changing how we see life itself.